very good day to everyone. So I'm Dr. Azlina, and today we're going to take a look at another physiology lesson, which is on the hematology, on the constituents of the blood. Okay, so without we go further details into the content, let's take a look at the learning outcome first, so that when we go through the lesson, you know which you need to grab. So which part is actually the important points in the constituents of the blood. So let's take a look at the number 1. First and foremost, you need to list of the components of the blood and what are the importance and what are the functions of the components in the blood. Okay, now let's take a look at the second point. So you should be able to describe the physical properties as well as the chemical components of the blood. On the third point, we're going to take a look at the hemopoiesis and the importance of the erythropoietin. I'm not going to touch details on the hemopoiesis because you're going to learn it in another chapter on the formations of the blood. And then we're going to outline the chemical components of the normal hemoglobin variant. So now let's take a look at the importance of learning the blood physiology. So basically, our bodily dysfunctions can be reflected in the blood. That's why learning the blood physiology is really important. And then a second one, why does we do take blood to assess a certain disease, to do a chemical or biochemical changes in the body system? Is actually because the blood is easily accessible. So means that it's easy to be taken out. So with just a finger prick, you can get a drop of blood, and it is actually a very simple biological samples that you can get for the investigation. Okay. When we take a look at the hematology as a general, so what is actually the hematology? So look at its name. Hema means the blood, logy means a study. So it means that we're going to study on the blood itself, the physiology of the blood, and as well as the blood disorder. Okay, so the study of the blood physiology will include the study on the different components of the blood. Okay, so what are the different components inside the blood? We're going to take a look at it later. Their formation, right? So how does this blood being formed, where does it originated from, and then their roles in the different body functions and their dysfunctions. Study of the blood disorder, we have the pathophysiology of the disease of blood. So we also can get the management on top of the blood disorder. And then there are also the pathological alterations in the blood, including non-hematological diseases. It's not necessarily changes in the blood structures or in the nomenclatures of the blood itself, but sometimes another dysfunctions of another body system can also be reflected, can also be examined or determined by obtaining the blood as the biological samples. So its fundamental aspect, if you take a look at the bone marrow, so bone marrow is actually producing the adequate number of the matured cells or the matured hematopoietic lineage. It is should be qualitatively normal. Quantitatively normal and qualitatively normal. So what does that mean by the adequate number? So it means that the quantity, the amount of the red blood cells in our system must be adequate, a number. There is a number that we should meet in order to perform the effective function of the blood, and as well as the quality of the blood itself. If let's say you have the normal number of the red blood cells, for example red blood cell, we're just focusing on the red blood cells, but somehow the red blood cell itself undergo malformation, so it means that the structures is not as what it's supposed to be, as what it's supposed to turn out like. Okay, contohnya dia adalah bentuk bulat, tiba-tiba dia jadi bentuk slender ke, oval ke. So means that one is not in a quality mode. Okay, so it should be quantitatively and qualitatively normal. Okay, so there are several investigations or hematological investigations that we can use in using the blood. So basically we have the differentials blood count. It could be smear examination. So this is examples of the diagram here shows the examples of the blood investigation. So on the right side here showing the blood count, okay, different blood count. And we can also study on the bone marrow. So means that the marrow, we do the bone marrow aspirate, and we smear the bone marrow, and then we look at the stem cells inside the bone marrow. stem cells are producing the blood from the bone marrow. Okay. So now let's move on to the compositions of blood. Contohnya kita withdraw blood ataupun kita prick, do a finger prick on top of the glass slide, and then there is 1 drop of blood or 2 to 3 drops of blood. Okay, so what are actually inside the blood? So that is what we call as the compositions of the blood. So the blood basically consisting of the cellular component as well as the fluid component. Of course lah kan, fluid component sebab dia cair. So when we come to the cellular component first, alright? So in the cellular component, you will see that inside the whole blood, okay, keseluruhan blood tu kita panggil whole blood, there are red blood cells, which we call it as the erythrocyte. There are also white blood cells, which is a leukocyte. There is a platelet, alright, a thrombocyte. Alright, so red blood cells tu red blood cells lah yang kita tengok red tu. White blood cells actually there are several numbers of red blood cells inside it. Alright, so white blood cell tu cuma general nama sahaja, leukocyte, but dia ada lagi variant dekat dalam white blood cells tu. And then we also have platelet, the one that cause the darah beku. Okay, now let's take a look at the fluid component. So inside the blood there are actually a plasma, okay, by which it contribute 55% of the total blood volume. It's actually made up of water as well as the solid particles. So apa solid particles ni? So other than the cellular component is actually the solid particle. Nanti kita akan tengok the solid particles tu apa kat dalam dia. Okay, so kebanyakannya adalah plasma lah. Alright, so look at this diagram on the left side. You will see that when you do a blood smear, yang kita buat blood smear yang macam ni kan, kita smear the blood on top of the glass slide, you will see that are a distributions of the red blood cells kat sini. And then you can also see the neutrophils, which is one of the white blood cells, lymphocyte, one of the white blood cells, and there are also the distributions of the platelet inside the blood smear. Okay, and from the whole blood, ni kita panggil whole blood lah. When you do a centrifugation, for example, kita masuk dekat dalam centrifuge and then we go centrifugation, but dengan syarat you collect the whole blood inside the EDTA tube lah. EDTA tube tu maksudnya dia ada anticoagulant chemical inside it, and it will prevent the blood from clotting. Basically kalau awak tengok tube tu warna purple lah. Dia punya tutup tu warna purple, so dia ada EDTA dekat dalam tu. Itu yang akan prevent blood from clotting. When you do centrifugation, you will see that packed red cells will be at the bottom, okay, after centrifugation. So when you do centrifuge, all of this blood has been packed down there. We call it as the packed red cell. And then on top of it, you will see like a yellowish fluid on top, and this is actually the plasma. In between of the plasma and the packed cell is actually the accumulation of Buffy coat, and in this Buffy coat consisting of white cells as well as the platelets. But we can say the plasma and the packed cell are separated by this Buffy coat that consisting of white cells as well as the platelet. Okay, what is actually inside this Buffy coat? Jom kita tengok. So inside the Buffy coat, as I mentioned, there is white cell, leukocyte ni white cell lah, as well as the platelet or thrombocyte. Right, so we get on one of the example. For example, like those who having systemic lupus erythematosus, primitive red cells, by which the red blood cells does not appear morphologically normal. Alright, metamyelocyte, blast cell, megakaryocyte, etc. So abnormal cells are found in the Buffy coat. Sebab tu kalau kita nak tengok disease ke apa ke, you buat centrifugations on a whole blood, okay, and then you ambil dekat Buffy coat dia, you smear that Buffy coat, you tengok structure dia macam mana. Okay, so other than that, for that Buffy coat preparation, you can use to detect the bacteria, fungi, parasites, neurophyte, monocyte, and other circulating macrophages in it. Okay, so here is the several functions of the blood. We're going to take a look at it one by one in details. So now let's move on to the functions of blood on the respiratory functions. So I bet that everyone know this already, okay, since you were born. Nanti cakap lah, ni kecil-kecil pun dah tahu kan? Yes, it is, because the first, blood is very important because it going to assist the respiratory functions, by which we know that the red blood cells are going to carry oxygen from the atmosphere and then bring to your lungs and undergo the process of gaseous exchange, and it really helps in the metabolic process of the system. Okay, so it's play roles in the respiration. Okay, so and then we take a look at the second point, by which the blood is actually served as a transport medium. Okay, so because of why? Because some of the hormones, okay, various hormones lah, most of the hormones are actually from the endocrine gland, from the tissues, or from the another glands, okay, adrenal medulla ke, all of these hormones are circulating in the blood. That's why we say blood is actually serving as a transport medium. They are also served as a chemical transport systems, okay, chemical substance transport system. And other than that, when you eat, all of the nutrient going to be absorbed in the blood circulation and going to be supplied to the whole system from the GI tracts and whatnot. Also the source of the vitamins, and then from the nutrient that you have absorbed to be circulated to the whole body system, this blood going to be used to regulate the growth and metabolism of the tissue. Now let's give one examples on the functions of blood on the temperature regulation. So let's give one examples. Like if let's say at the end of your exercise, basically after jogging, after doing a strenuous exercise, you might notice that your face might turn out a bit red and your skin's going to be a bit hot, right? So why is that so? That is because our blood system are basically like a coolant systems. It's like a coolant system in a car, for example. It's just a similar things, alright? But somehow this is in a biological system. So our blood, basically, when we do some sort of that kind of exercise, they are actually in a sympathetic response. So the blood vessels going to be vasodilated. So it will be close to the skin and it will be increased in diameter. Why? Because it going to dissipate the heat that being produced by our body system. So they're going to undergo vasodilations and heat dissipate out from the body into the environment so that our body can be contained to the temperatures of 36.5 and 37.5. Okay, so normal temperature. But then, if let's say you are sitting in a cold place, for example, you're going to Cameron Highland or something, you will feel like your hands a bit kebas, a bit cold, and then turn out to be a bit blue. So why? Because your blood vessels during that time are under vasoconstrictions, alright? So it's going to constrict, reduce in the diameter, because why? Because it don't want to dissipate the heat out of your body, and it wants to conserve the heat inside your body to preserve your core temperature body system. So that is the temperature regulations, the functions of the blood. Now let's take a look at the next functions by which the blood used to excrete a certain metabolic waste from our body, and basically these functions are carried out by the kidney. And we notice that kidney, if you take a look at its structure, is actually it is actually red in color as well, right? But somehow inside the kidney there are a lot of blood vessels inside there in the glomerulus, by which its purpose is to excrete the waste materials, the waste product inside the blood itself. So our blood not actually bring nutrients, it also carry a waste product. So where its destination? So the destinations for the waste product is actually to the kidney, by which kidney will help to filter and get rid all of the waste substance into the urine. Now let's take a look at the water homeostasis. As I mentioned earlier, inside our blood, 55% of it contain plasma. So out of that 55%, the rest of it, 92% of that plasma is actually a water. Okay, so that's why if let's say you see a people that having hypovolemic, for example, hypovolemic stress means if let's say they are in a extremely hot weather for so long, their body start to be dehydrated. So when they start to dehydrated, the blood going to be so viscous and it cannot carry out its functions properly. So that condition we call it as the hypovolemia. So how to treat the hypovolemia? Tadi kita bagi contoh hypovolemia. Another situation is actually like diarrhea, alright, vomiting, excessive sweating ke macam mana ke. So that can contribute to the loss of water. So macam mana kita nak counter balik ni yang loss of water tadi? Sebab tu kita masukkan IV drip, and then kita bagi contohnya those who having diarrhea, we treat them by giving them the ROS, right, oral saline solution, so that they can conserve back the water from another extracellular body fluid into the bloodstream. Okay, so other than that, in a conditions like hypovolemia, okay, means that hypovolemia means hypo to reduce volume in the blood. Hypo tu kurang volemia volume. Okay, so in the conditions of the hypovolemia, it's basically will activate a system that will conserve back the water so that it can maintain the blood volume, because 5 liters of blood volume in a normal body weight is actually very optimum amount of the blood to carry out the functions. If let's say you reduce in a volumes of that blood, it cannot carry out the functions properly. Okay, blood is actually to serve as the acid-base balance. Alright, as I mentioned earlier, part of the constituents of the blood is actually a protein, right? So what are the protein inside the blood? Is actually one of it is actually the hemoglobin. So as we all know, hemoglobin not only serve in the gaseous exchange as to carry oxygens, but also to carry carbon dioxide, and other than that, this carbon dioxide later, when it undergo exchange in the blood plasma after it been taken out by the tissue into the bloodstream, right, in the systemic circulations, this carbon dioxide going to be dissociate. Alright, it will be dissociate and it will form the carbonic acid and also the hydrogen ion. And hydrogen ion, as we all know, it is actually the H plus ion. So H plus ion is basically acidic kind of ion. So when it is acidic, it need to be conserved and it needs to be buffer so that it won't affect the other body system, change the pH of our body system. Therefore, where does this H plus going to go? Going to be taken up. So it will be taken up by the hemoglobin. So jadi HbH plus lah, hemoglobin lah. So as what we can say, the blood is actually serve as a buffer system. Okay. Alright, the next is actually to serve as the immunity. So we believe that in our blood we have the immune system. Alright, we call it as the immunoglobulin. Right. So immunoglobulin is actually coming from the blood. It contains antibody inside our cells, and it's actually a protein lah. Immunoglobulin is actually a protein that will to serve as the immunity system. And the rest is actually as a storage for the nutrients, electrolytes, hormones, and then it's blood as well, give the body color. As I mentioned earlier, just now on temperature regulations, if let's say your body is too hot, it's turned out to be red. If you are too cold, it's turned out to be blue. Similarly, okay, the concept is applied to conditions by which a person having low blood content. Okay, this is examples of the anemia or hypovolemia. Their body tends to be a bit pale. So why? Because the heme color, right, from the hemoglobin is reduced. So nutritive functions, as I mentioned, it going to deliver nutrients. And then other than that is actually to serve an oncotic pressure. Okay, let's recap back on a topic on the blood volume. Apa yang osmolarity semua tu kan. Okay, chapter 1 atau chapter 2 macam tu on the fluids and regulations. So you belajar osmotic, you belajar isotonic, itu you belajar apa lagi? You belajar hypertonic, hypoosmolar, also osmolar, semua tu kan. So this is where you're going to apply. So oncotic pressure means that the pressure that is exerted by a solute. Solute tu maksudnya particles lah dekat dalam ni. Okay, kita tengok the left image first. So you have this cylinder is actually representing the blood vessels. Okay, and then you will have the plasma protein, the yellow one representing the plasma protein, for example like albumin ke, globulin ke, fibrinogens ke, whatever it is. Basically the albumin lah, because albumin contributes a majority of the proteins inside the blood circulation. So this solute actually going to conserve water. Dia akan bertindak sebagai kita bercakap di mana ada solute, dekat situ water akan ikut. So this is actually the concept. Where is the solute, the water will follow it. Alright, so this protein albumin inside the blood system are actually to serve as the oncotic pressure so that it can retain the amount of water inside the blood circulation. Okay, so sebab kalau air, dia akan buat hydrostatic pressure. Kalau kita ada solute, dia akan menyebabkan oncotic pressure. That's why the volume of solute to the volumes of water must be ideal so that it can conserve the amount of water inside it. If let's say a person having a problem, for example, dia ada masalah tak boleh produce albumin ke kan, contohnya siapa yang produce albumin tu? Yang produce albumin adalah liver. Okay, siapa yang buang pula albumin daripada badan dia? Kalau orang tu ada kidney disease, that albumin going to be secreted out by the kidney, so they will have a reduced volume of the protein, therefore it cannot conserve water inside the body. That's why kalau you tengok kalau orang tu ada masalah kidney disease, for example kidney failure ke, badan dia akan bengkak-bengkak. So why? Because most of the protein in the blood circulations are excreted out by the kidney, jadi dia tak boleh nak conserve water, so this water inside the blood vessel dissipate out and causing the edema in the surrounding of the body. So that is the functions of the blood itself, the protein, the constituents of the blood, which is a protein in the blood circulations. We done on that on the functions. Now let's move on to the blood volume. Okay, so basically in adult, male ke dan female ke, kita kata randomly our blood is actually 5 to 6 liter. Right, basically male would have a larger amount in the blood volume comparing to the female, is due to the body size, and of course if let's say a children, they will have significantly less amount of the comparing to the adult. Right, so how do we determine the blood volume? We determine the blood volume by the ratio of the plasma to the cell volume. So the plasma to the cell volume is actually 55-45. So as I mentioned, water is constituting 92%, and the solid or other solids, for example like plasma protein, will make 90% of that solid. The other solid and solute is actually 8%, and out of this 8%, 90% dia adalah plasma protein. Okay, so kita ada inorganic as well as organic solid. So organic is actually the plasma protein, glucose, lipid, urea, uric acid, and another inorganic is actually the ion and also cation. So how do we measure the cell volume? How do we measure measurement of the cells volume inside the whole blood? Okay, so the volumes of leukocytes and platelet basically are going to be ignored. So apa yang kita measure kalau kita measure cell? As I mentioned earlier, dalam slide yang pertama tadi, I did mention we have a cellular component and we have a water component, fluid component. That cellular component coming from, you remember, it's coming from the leukocyte, platelet, and the majority of it coming from the red blood cells. Red blood cell, white blood cell, and platelet. Leukocyte ni platelet, white blood cell lah. Alright, but then the leukocyte and platelet is very minute, therefore we ignore that cell volume and we just calculate the red blood cells. Alright, so the volume occupied by the circulating cell lah. Kalau kita cakap red cell volume adalah volume occupied by circulating red cells. So how do we measure? We inject the red cell using radioisotope, and then we calculate using that one lah. Alright, so the dyes that we use basically even blue dyes, and we also measure using injection of serum albumin labeled with the radioactive iodine. Okay, so this is the method that we use to calculate. Maybe you can try to calculate this one. Now let's take a look at the effective blood volume. Effective blood volume is actually volumes of the whole blood you minus with the volume of the sequestrated. So what does that mean by sequestrated? So sequestrated is actually the amount of the blood that is dissipate or squeeze out of the blood vessels. So means only blood yang ada dekat dalam blood vessels tu saja we call it as the effective blood volume. Okay, so means the volume that presents in the circulations. Ni saya dah cakap kat sini the volume that presents in the circulations. So now let's take a look at the specific gravity and viscosity of the blood. Looking at the term specific gravity, so kita tahu specific gravity means that it's going to tell you about the gravity of a certain things. Yes, it is true specific gravity. And then the viscosity of the blood, so means that the compactness of the blood, the kepekatan dia. Viscous lah, kepekatan blood tu sendiri. Alright, specific gravity or density of the whole blood is actually approximately 1 lah. We consider as 1 point, is actually 1. But how it is depends on. So it is actually depends on the number of the cells as well as the compositions of the plasma. If let's say a person is having loss of blood due to dehydration ke, hypovolemic ke, kan apa-apalah basically dehydration lah, due to the loss of water, not loss of blood, loss of water, it will increase the specific gravity of a blood. And when the specific gravity is reduced, so the viscosity going to be increased. And then why do we talk about the viscosity? Because viscosity had an influence on the blood flow. Okay, so the viscous the blood is, the increased resistance of the blood flow. So means that kalau dia increase resistance, maksudnya dia susah nak bergerak. Macam you ada minyak dengan you ada air, and then you salurkan dekat paip, yang mana lagi laju? So air tu lagi laju comparing to the minyak kan. So that is actually a simulations for a viscosity of the blood lah. It will increase the resistance. Blood viscosity here is mentioned about 4 to 5 times of a water. So water tu macam cair betul kan. So blood punya viscosity, kepekatan dia, kelikatan dia is 4 to 5 times than water. Now let's move on to the compositions of the plasma. So as I mentioned earlier, plasma contributes around 55% of the whole or total blood component. Alright, so please bear in mind that there are two things. We have plasma as well as we have serum. So what is actually the difference between plasma and serum? So plasma is actually the one that is collected. Okay, we collect a blood and then we put it inside the EDTA tube, which means that the tube, usually purple in color, cap that consists the anticoagulant in it. So we withdraw that blood and we store it in the EDTA tube, by which in that EDTA tube, the anticoagulant will prevent the blood from the platelet to form a fibrinogen and to form clot. Okay, so that is actually the plasma. But if you want to take a serum, you take a blood and then you put it inside the plain tube, the tube that doesn't have any anticoagulant in it, and you store it in 2 hours, and then it will clot. Okay, so when it is clot, the one, and after that you centrifuge, the one that you get is actually the serum, the blood serum. Alright, so blood plasma is actually the one that is collected with anticoagulant. Usually we can use that blood plasma for the investigations, for blood investigations as well. And for the fresh frozen plasma, FFP, it can be stored below -20 degrees. To preserve the clotting factors, the plasma should be frozen within 6 hours after collection. Inside the plasma, there is a clotting factors lah in it, because this does not clot, so the clotting factor is still there. But somehow if you want to preserve that clotting factor, kena cepat-cepat lah, within 6 hours after collection dah kena simpan dah. So we use that for the plasmapheresis lah, untuk kita certain patients dia perlukan plasma je, so jadi kita ambil plasma ni lah. Alright, so this is actually the composition of plasma, but you can see 55-45. If what you can see, protein contributes around 7%, and you have water 92%, and another solute is actually 1%. So what are the protein inside the plasma? Is I mentioned earlier, we have the albumin. Majority of the protein is actually the albumin. You have a globulin, fibrinogens, and then the others is actually 1%. And all of these protein are actually serve as the oncotic pressure. Saya cakap tadi kan, yang ada oncotic pressure, yang pressure nak conserve the water inside the mucosa, right? Alright, so we have a water 92, and the other solute, what are they? They are electrolytes, nutrient, waste product, regulatory substances like hormones and whatnot. Okay, so all of these are actually belongs to another 1% of the plasma constituent. This is the serums that I mentioned earlier. So serum is actually, that is a plasma, but plasma without a clotting factor, because kita dah let it to clot kan, jadi clotting factor tu dah clot, jadi yang tinggal hanyalah serum sahaja. Okay, so this is the difference between the in a purple cap is actually the one that contain the anticoagulant. Masa you collect tu, you put that blood into the anticoagulant tube, and then you centrifuge, you will get this blood plasma that contain a fibrinogen inside it. You will have a Buffy coat and red cells. But if let's say you collect the blood and you put it in a plain tube, okay, and you let it for a while, let it to clot, and later you undergo the centrifugation, and after that you will see that on top is only a clear fluid. We call it as the serum, because this one no longer having a fibrinogen, and down below is actually the clotted blood. Okay, so this is the different lah. Alright, so kalau if you take a look at this purple one, you will see that there is the platelet in between or inside the Buffy coat. Because why? Because the platelet does not being used to undergo the clotting. But for this one, there's no longer platelet inside here because the platelet has been used to clot. Okay. So we have 3 types of plasma protein. Okay, and basically all of these plasma protein are being developed or originated or prepared by the liver. Okay, formed in the liver, and they are albumin, globulin, and fibrinogen. As I mentioned earlier, albumin is actually the majority of the protein in the plasma. Okay, now let's move on in details to the each of the proteins we actually mentioned earlier. So we have the albumin, and albumin is actually, its half-life is 20 days, and its molecular weight is actually 66. If you take a look at the diameters of the albumin, it's actually not a big protein, but somehow it is majority of the protein is there. Maksudnya dia banyak kat situ, tapi dia bukanlah protein yang terbesar kat sana. So it has the smallest diameter, and if let's say a person having kidney disease, as I mentioned earlier, this albumin can be get rid of into the urine. So kalau orang tu kencing, and kita ambil air kencing dia, kita test, kita tengok ada albumin, so means that must be something wrong lah with the kidney, because supposedly kidney does not excrete the albumin, and that condition we call it as the albuminuria. So means that albumin that present in the urea. Okay, so means that those that having albuminuria, maksudnya albumin tu dah dibuang ke dalam urea, ada lagi tak albumin dekat dalam blood? So kuranglah albumin dekat dalam blood. So that condition, in the blood condition, kita panggil hypoalbuminemia. Okay, so maksudnya dia dah kurang albumin dekat dalam blood, because albumin tu ada dekat dalam urine. Okay, so it's not necessarily those who have albuminuria ataupun hypoalbuminemia ni adalah dia ada kidney disease, because hypoalbuminemia ni can also occur in a liver disease, by which that liver cannot produce enough number of the albumin, low productions of the albumin. Alright, so bila dia dah albumin dah kurang, jadi colloidal osmotic pressure pun akan kurang dan menyebabkan orang tu ada edema. This is the one that I mentioned earlier. Bila albumin tu dah buang, maksudnya dalam tu ada air saja, dia tak boleh nak distract water masuk dekat dalam blood vessel. So water tu akan dissipate out from the blood vessel, so dia akan pergi dekat surrounding, menyebabkan orang tu jadi edema lah, bengkak-bengkak kan. Okay, so now let's move on to the next protein, which is the globulin. So we have alpha, beta and gamma globulin kalau you tengok kat sini, and I mentioned produced by the liver, and this globulin basically used as a transport protein. Okay, so we have a transferrin, cellular plasma protein. So this is a transport protein. So basically for the transport protein, dia akan embedded in the plasma membrane. Okay, dekat membrane dia dia jadi macam channel lah. Okay, and then we have lipoprotein. When it's combinations with the lipid, we will form the lipoprotein. So lipid mean lipid lah, protein lah, so jadi lipoprotein. And then part of the globulin is actually the immunoglobulin, the one that you use as the antibody lah. Basically that type of protein is the gamma globulin. Alright, so normal albumin to the globulin ratio, we measure it as AGR. We call it as the AGR, and this AGR basically used to measure a state of a certain infections or diseases. For example, like immunological reactions or inflammatory disease, you will see that if you take that blood and then you see that the AGR, that person having a lot more of globulin, okay, comparing to the albumin, so means that the person is having infections or inflammation. Maksudnya dia tengah banyak lah the mass production, sebab dia nak counter balik inflammation tadi, sebab tu lah banyak ratio of the globulin in comparing to the albumin. Okay, next is actually the fibrinogen. Nama pun fibrin. Fibrin maksudnya dia akan buat untuk coagulation lah. Dia akan mesh jadi coagulation. So because of the fibrinogen ni, dia macam long, tangling kan. So jadi dia akan contribute to it will contribute to the viscosity of the plasma, and as it contribute to the viscosity, it will use to determine the ESR. So what is ESR? ESR is erythrocyte sedimentation rate. Erythrocyte sedimentation rate tu apa? Dia sediment lah. Maksudnya dia akan, bila you, contoh I give you a simulations by which you ada air tanah, air yang lumpur kan, you put in a bottle, you put a soil in it, you put a water, and then you shake, and then you leave it for a while, and then you tengok dia sediment dekat bawah berapa lama. How long does it take to settle at the bottoms of the bottle? So similar to this condition, okay, to this the one that we are talking about in the ESR, by which this fibrinogen are basically going to influence the erythrocyte sedimentation rate. So dia yang akan influence red blood cell ni akan sediment dekat bawah. Okay. So apa fungsi-fungsi nak sediment kat bawah tu, nanti kita akan tengok lah erythrocyte sedimentation rates tu. What does that mean by the erythrocyte sedimentation rate? Okay, so this is the functions of the plasma protein. At the beginning, we talk about the functions of the blood as a whole. Now is the functions of the plasma protein. So let's move on to one by one. The osmotic pressure, as I mentioned, is contribute to the oncotic pressure, and for the oncotic pressure, is basically 25 millimeter of mercury. Alright. Okay, so this one is the viscosity of the blood. Okay, is it important to get to know the viscosity of the blood? Plasma protein contribute to the viscosity. Yes, it is really, really important, because if your blood is too thick, it's very hard to deliver the blood to the systemic circulation, because it will increase the resistance. So blood tu tak boleh pekat sangat, tak boleh cair sangat. Right. And then what are the things that contributes to the viscosity is the fibrinogen, because it is elongated and fibrillar in structures. Okay. Okay, for the immunity, I did mention earlier. Okay, coagulations as well. This the ones that I list here is actually I already mentioned. It's just for your own to do your revisions during your study period lah. Until you study baru you baca kan. Okay. Transport protein, as I mentioned, it will transport hormone, drugs and metals and etc. Because hormone, kat mana hormone tu dia disirkulatekan? Dia diproduce dekat tempat lain, tapi dia dihantar dekat tempat lain to mediate C actions kan, to stimulate C action. So what is the channels that carry hormone? Is actually the blood circulation. So blood tu adalah transport medium dia untuk transportkan hormone ni to the whole system. Okay, so buffering I did mention earlier, because it serve as a buffer system to homogenate, to what do we call, not homogenate lah, macam to equalize balik the body pH, and then the protein store, because it store a lot more protein in it, and then for the synthetic function to synthesize a protein hormone like erythropoietin. So in the blood plasma, it provides substrates for synthesis of erythropoietin. So erythropoietin ni produce dekat mana? So please jot down there, erythropoietin is produced in the kidney. Alright, so now I look back on the erythrocyte sedimentation rate. Tadi saya dah bagi tahu, the fibrinogens tu is used to determine the erythrocyte sedimentation rate. Erythrocyte sedimentation rate is actually depends on the concentrations of the fibrinogens that available in the plasma. So why? Because the fibrinogen is actually going to facilitate in the relux formations. So relux formations tu apa? Ni, yang staking of the red blood cells ni. Because this one, when it's taking into the red blood cells, it's influence the viscosity of the blood. So the relux formations, the formations of the relux itself will increase the sedimentation rate of the red blood cells. There are various methods that we use for the separations of the plasma proteins. Either we use as a salt preparations, electropoiesis, confractionation, ultracentrifugations, or immunohistochemical analysis. So to determine the concentrations of various fraction is very useful to estimate the AGR and for the diagnosis and also the prognosis of different diseases. Now let's move on to the hemopoiesis. I will not touch this hemopoiesis too much because you will learn this in another topic, but somehow just to have a quick recap on the hematopoiesis or hemopoiesis, it actually the process that occur in the spleen and liver in a fetal life, right? And then it's occur in a red bone marrow, and from the childhood when you are growing up, this red bone marrow going to be replaced by the fat tissue, and later we call it as the yellow marrow instead of red bone marrow. So in adult, so basically this process occur in all of this stated region. So bone marrow is actually normal size for hemopoiesis, or sometimes you call it as the hematopoiesis. So active process of producing the red cells, white cells and platelets, and it's actually increase in demands when a person is having hemorrhage or infections. So dia akan lagi trigger bone marrow ataupun the regions that producing the blood cells to be activated or to be stimulated. So what are the things that trigger the productions of the red blood cells or the blood cells in the body? It's not only the condition itself, but it is actually being called by a certain substances which is known as the erythropoietin. Erythropoietin ni lah macam sauce dia. Bukan sauce tu sauce ni macam apa, emergency call. So erythropoietin is produced by the kidney, by which when the blood in our body is circulating inside the systemic circulations and it reach to the kidney having cells that sense the concentrations of the blood cells inside the body circulation or systemic circulations. Bila dia sense, dia hantar scanner, and then scanner cakap, oh tak cukup lah you punya blood component dekat dalam you punya circulation. So the kidney will start producing the erythropoietin, by which this erythropoietin is not going to tell the kidney itself, dia yang produce, tapi dia akan bagi tahu dekat, dia macam operator lah, kidney ni operator producing call, produce erythropoietin circulated in the blood system, pergi bagi tahu dekat tempat yang produce hematopoiesis or hemopoiesis to produce a lot more of red blood cells to replace. Okay. So there are several factors that promote erythropoietin secretions. For example, like hypoxia, alright? Hypoxia maksudnya kurang oksigen lah. Okay, alkalosis or hormones, and the other hormones like growth hormone, prolactin, thyroid hormone, or hemolysis lah. Hemolysis ni maksudnya certain things that lyses the lyses the red blood cells. Okay, I think that's all for the topic today. Hopefully you all can digest on the content. If let's say you have anything to ask, please feel free to contact me. Thank you for your attentions, and see you again next time in the next chapter. Assalamualaikum.